Living in this community is about communication, collaboration, and problem solving through critical thinking. We tested these when we encountered the challenges creating this project, including teammate disagreements and technology difficulties. To overcome these problems, we communicated with one another and collaborated to come up with a plan of steps on how to resolve the disagreements and work towards our goal. Leading up to the project, we began by setting up a game plan, discussing project rules while gathering research, and contacting our sponsors.
Main objectives of this project are to work on technical, problem-solving, and communication skills, making us more competitive when job searching. During the project, we practiced using the software Rhino as well as the laser cutter. With Rhino, we struggled with the shortcuts on the program, and with the laser cutter, we had difficulty calibrating it. But to resolve these problems, we asked ILT staff for help in the studio. And before cutting the final piece, we made sketches as well as a physical prototype. We emailed our project coordinator about where to find materials, and he provided guidance throughout the project. As a group, we communicated almost daily to schedule meetings, and our project mentors were a big help with communication and making sure everyone did their part.
Rumen is the largest section in a ruminant's stomach, and it is the first stop during digestion. Microorganisms and bacteria contained in this section break down food particles through fermentation. The next stop food makes is the reticulum. The reticulum is sometimes known in conjunction with the rumen as the reticular-rumen complex due to the lack of a wall separating the two complexes. The reticulum has a honeycomb structure and is used as a chamber to trap and digest dense material that the animal has consumed.
The omasum is made of many pleas, which are leaves of tissue that allow it to maximize absorption. It also reduces the size of heavier particles from the reticulum before they enter the abomasum. The abomasum is the glandular stomach of ruminants, where food is completely broken down by the churning action created by the contraction of stomach muscles. It also secretes hydrochloric acid for chemical digestion and can be used to store food before it goes onto the small intestine.
The stomach is an essential organ of the digestive system that aids in both mechanical and chemical digestion of food consumed. This is achieved by muscular contraction of the stomach walls and secretion of gastric enzymes and acids.
The muscles of the stomach only contract in one direction. Thus, there are 3 layers striated in different ways. As these muscles contract and digestive enzymes from the mucosa are secreted, the stomach breaks down food into substances that can be absorbed by the body. Once it is ready to be absorbed, the pyloric sphincter aids in the control of the movement of chyme into the small intestine.
The most obvious difference between the two structures is their actual physical appearance and organization. There are 4 parts to a human stomach, whereas there are literally 4 different chambers in the ruminant stomach. Another difference between the two digestion structures is the digestion process itself. A human stomach uses enzymes and muscular contraction to break down food into a chyme to be further digested in the small intestine. The ruminant stomach uses bacterial method rather than enzymes and breaks down food in 4 different phases in each of the 4 chambers. Some similarities between the two stomachs: both human and cow must utilize mechanical and chemical digestion methods to process the food and break it down into absorbable monomers. And also, both use hydrochloric acid to break the food down.
So over the course of this project, our group was able to create a successful product. We ran into some issues along the way. We had some trouble getting all of our group together to do the project every week because of our differing schedules. We were able to overcome this by realizing that we could each do parts of the model individually or as smaller groups. Ultimately, we were able to use our individual skills to make our team better. We grew as parts of the CureVinci community through this project. We got closer to each other and learned more about working as a team.